Hello and a very good day to everyone. My name is Joshua Low Junyi, a student in the Masters of Science in Molecular Medicine, and today I'll be presenting on a pivotal tool in modern healthcare, vaccines. Over the next few slides, I'll walk you through the history and principles of vaccination, delivery strategies, current challenges, and exciting emerging innovations. Let's start off with the introduction. Vaccines are arguably one of the greatest public health achievements in history. As biomedical tools, they work by stimulating the immune system either with weakened, inactivated, or a specific component of a pathogen to develop antibodies and memory cells for long-term protection. According to WHO, vaccines have prevented over 30 life-threatening diseases and save up to 5 million lives annually. They also contribute to herd immunity, offering indirect protection to individuals who cannot be vaccinated. Diseases like smallpox have been eradicated, and others like polio and measles have seen dramatic reductions due to the widespread immunization. Vaccination is not a modern concept. Its roots trace back to the 15th century in Asia and Africa, where variolation has been practiced. The modern-day era of vaccine began in the 18th century by Edward Jenner. He demonstrated that inoculating individuals material from cowpox lesions could confer immunity to smallpox. This experiment with an 8-year-old boy marked the birth of modern immunology. The development of vaccines continued rapidly through the 19th and 20th century, where Louis Pasteur created the first live-attenuated vaccine for rabies, to the development on inactivated polio vaccines in the 1950s. In more recent years, we've seen milestones such as the global eradication of smallpox, introduction of the HPV vaccines for cervical cancer prevention, the first Ebola vaccine using the viral vectors, as well as most recently the mRNA vaccines like those used against the COVID-19. Vaccines can be broadly categorized by how they introduce antigens to the immune system. Let's review some of these major types. Live attenuated vaccines use a weakened form of pathogen where their virulence have been reduced through genetic engineering. They induce strong immune responses, including both humoral and cell-mediated responses, but are not suitable for immunocompromised individuals. Examples include measles and mumps vaccines. Moving on, inactivated vaccines are another form of pathogen-based vaccines where they contain killed pathogens. They are safer but often require multiple doses and adjuvants like the inactivated polio vaccines due to the establishing moderate humoral responses and weak cell-mediated responses. Next, recombinant vector vaccines use harmless viruses to deliver genetic materials from the target pathogen. This avoids the risk of live vaccines where a good example includes the Ebola vaccines. Recombinant vector vaccines are classified as replicating and non-replicating, while replicating vectors can multiply within the host cells and elicit strong humoral and cell-mediated immunity, while non-replicating vectors do not replicate and primarily induce humoral responses. Nucleic acid-based vaccines include DNA and RNA vaccines represent a recent advancement in vaccinology, garnering increased pharmacological attention due to the COVID-19 pandemic. These vaccines instruct host cells to produce antigens internally, which are then processed and presented by the MHC molecules. These are fast to manufacture, versatile, scalable, and can induce both humoral and cellular immunity. mRNA-1273 is a notable example where it's an mRNA vaccine that encoded the SARS-CoV-2 spike protein. As illustrated here in Figure 2, mRNA vaccines are generally preferred over DNA vaccines due to their more efficient cytoplasmic delivery, faster protein expression by bypassing the nuclear entry, and a lower risk of integration into the host genome, resulting in a safer and more rapid immune response. Lastly, let's take a look at protein-based vaccines. Subunit vaccines use purified proteins or recombinant antigens derived from the pathogen. These proteins stimulate an immune response without introducing the whole pathogen, making these vaccines very safe since there is no risk of causing the disease. Nevertheless, they are less immunogenic and require the addition of adjuvants to enhance and prolong the immune response through immunity. Viral-like particles are another type of protein-based vaccines where they mimic virus's structure but lack the viral genetic material, so they cannot replicate or cause the infection. They are composed of viral proteins that self-assemble into particles resembling the native virus, which helps the immune system to recognize them effectively. This stimulates strong immune responses as seen by the Gardasil 9 for HPV. Let's now take a look at vaccine delivery strategies, which are key to optimizing efficacy, safety, and immune response. The chosen route of delivery directly impacts how well the vaccines induce immunity and its safety profiles. There are two main categories, the parental route and mucosal route. When administered in the parental route, antigens form depot in the injection site where they gradually drain into the nearby lymph nodes where they effectively activate the immune system. Among these, subcutaneous and transdermal routes may provide better lymphatic drainage and subsequently stronger immunogenicity. However, IM injections are generally considered safer due to less risk of local reactions. For mucosal routes, this involves delivering vaccines through the mucosal surfaces. For these vaccines to be effective, the antigen must survive the harsh environment of the gastrointestinal tract or the nasal passage. Examples include the flu mist, an intranasal influenza vaccine, and the oral polio vaccine, both of which stimulate immunity right at the pathogen's point of entry, providing effective mucosal protection. A well-designed delivery system also enhances immune responses by improving several key factors such as antigen viability, efficient cellular uptake, metabolic stability, and precise targeting of relevant tissues. This is especially important for non-whole pathogen vaccines. The most basic delivery system includes the naked antigen delivery, which involves formulating the antigen simply in a buffer and directly injecting them. While that being cheap and straightforward, it suffers from rapid degradation in the body, poor targeting to immune cells, and consequently low immunogenicity. Mechanical delivery systems, howbeit, physically propel DNA-coated particles into cells. However, they can damage cells and generally exhibit low efficiency with variable antigen uptake. Moreover, viral delivery systems, including the viral replicon particles, are engineered viruses designed to infect cells only once, where they deliver antigen-encoding genetic material but cannot replicate or spread, ensuring safety. This strategy induces robust immune responses due to the efficient intracellular antigen expression. However, they are relatively expensive and face limitations due to their body's immune response neutralizing the vector upon repeated use. Hence, other non-viral approaches like nanoparticles have gained significant attention, offering versatile platforms to protect antigens from degradation, enhance uptake by immune cells, and enable targeted delivery to lymphoid tissues. Now let's take a closer look at Figure 4, which illustrates the key structural components of several well-established nanoparticle systems used in vaccine delivery, including LNPs, polymer nanoparticles, cationic nanoemulsions, lipoplexes, and lipopolyplexes. These systems are engineered to enhance delivery efficiency by protecting the vaccine cargo, facilitating cellular uptake, and enabling intracellular release. They are particularly important for delivering fragile molecules like mRNA, which require protection from enzymatic degradation and targeted delivery into the cytoplasm for translation. Amongst them, LNPs have gained the most attention and now the most widely used platform for delivering modern vaccines. They are uniquely designed to improve intracellular delivery by protecting fragile biomolecules, facilitating cellular uptake via endocytosis, and even acting as adjuvants to enhance immunogenicity. These functions are supported by the list of components in Table 1. Ionizable lipids facilitate the cellular uptake via endocytosis and promote endosomal escape in response to acidic pH. Phospholipids enhance biocompatibility, structural stability, and membrane fusion. Cholesterol contributes to the overall stability of the LNP and supports endosomal escape. And PEG lipids help control particle size and extend circulation time by reducing the opsonization and early immune clearance. A well-known example is the BNT162b2 mRNA vaccine, which uses LNPs to deliver mRNA encoding the SARS-CoV-2 spike protein, which enable efficient delivery into the host cells, triggering robust immune responses. Moreover, LNPs can be made biodegradable to improve biosafety and can be surface modified for targeted delivery. Nevertheless, vaccine development has stumbled upon various hurdles over the years. This diagram illustrates the multifactorial challenges that continue to hinder vaccine development, optimization, and population-wide effectiveness. One of them is our incomplete understanding of how the immunity develops, particularly for emerging pathogens. Some vaccines show poor immunogenicity, especially in certain populations. In many cases, we lack correlates of protection, clear biomarkers that predict vaccine-induced immunity. While immunologists have catalogued many of these individual components of immune system, we still struggle to understand how these elements work together as a coordinated whole. This lack of holistic insights means we cannot reliably predict immune responses to infections or vaccinations. There may also be an inadequate innate immune activation and the lack of reliable animal models, which further complicates preclinical studies. Host pathogen variability also remains a key challenge in vaccine effectiveness. Immune responses can vary slightly depending on individuals due to their genetic, demographic, and immunologic factors, with some individuals being non-responders who fail to develop adequate immunity. Sex, age, race, and ethnicity also influence the vaccine efficacy and safety. On the other hand, pathogen variability, especially in rapidly mutating RNA viruses like influenza, leads to antigenic shift, reducing vaccine effectiveness and necessitating for frequent updates. Some viruses are also hypervariable and capable of immune evasion, while others exist in latent active forms, further complicating host pathogen interactions and the development of long-lasting protection. Vaccine safety is also always a priority and a major public concern. Vaccines may occasionally cause adverse events, although most are rare and mild, and may potentially elicit autoimmune reactions due to molecular mimicry. With this, vaccine hesitancy remains a growing challenge globally. Nonetheless, emerging fields such as adversomics seeks to understand genetic and molecular factors behind adverse vaccine responses. Lastly, environmental and geographical factors also significantly affect their performance. Poor nutrition, obesity, co-infections, prior immunity from natural infections or previous vaccinations, and environmental pollutions may potentially all impair the immune and vaccine responses. Hence, overcoming these challenges are crucial to advance global health. This includes developing next-generation vaccine platforms offering the promise of enhanced immunogenicity, fewer adverse effects, multivalent pathogen targeting, rapid scalable production, and cost-effective manufacturing. Equally important is investing in more predictive animal models to better simulate the human immune responses. Besides that, the inclusion of diverse populations in clinical trials will ensure vaccines are safe and effective for all demographics, and involving multi-omics to evaluate vaccine efficacy by detecting unique molecular signatures, facilitating imprecise epitope mapping, identifying off-target effects, and enabling personalized vaccination strategies. Additionally, artificial intelligence are increasingly vital for predicting viral mutations, guiding vaccine updates before outbreak occurs. Lastly, developing a thermostable vaccine can simplify storage and distribution, especially in resource-limited countries. Penultimately, let's discuss on the latest advancements in vaccinology. Self-amplifying RNA vaccines and advanced subclass of mRNA vaccines are one of them. They incorporate a genetically engineered replicon, a sequence derived from viral replicase genes that encodes non-structural proteins responsible for RNA replication within the host cells. This design allows the RNA to amplify itself intracellularly, leading to a substantial increase in antigen expression. Consequently, comparable levels of immune protection can be achieved with a significant lower doses compared to conventional mRNA vaccines. This not only reduces the production cost and adverse effects, but also enables the possibility of single-dose immunization with protective effects lasting for months. Additionally, siRNA vaccines are produced via the in vitro transcription, a relatively cheap and scalable process which makes them highly suitable for rapid development during outbreaks. A notable example is the ARCT-154 developed to combat the SARS-CoV-2. Beyond infectious diseases, this platform holds immense potential for broader applications, including treatments for non-communicable diseases such as cancer. Figure 7 illustrates the mechanisms of siRNA vaccines. Upon cytoplasmic entry, host ribosomes first translate the 5' open reading frame to produce the viral replicase complex. The non-structural proteins assemble into a replicase complex that recognizes a promoter in the 3' untranslated region, initiating the amplification of genomic RNA and generation of subgenomic RNAs encoding the antigen. This leads to the high level of antigen expression, which are subsequently processed in two key immune pathways. In the MHC1 pathway, antigens are degraded by proteasomes and presented to the CD8 plus cytotoxic T cells, initiating a cell immune response. Simultaneously, antigens are endocytosed and presented on MHC2 class molecules to CD4 helper T cells, promoting B cell activation, cytokine secretion, generation of plasma and memory B cells, thereby inducing humoral immunity. Additionally, the replication process produces double-stranded RNA intermediates, activating innate immune sensors and further enhancing the immune response. This table summarizes preclinical and clinical trials of siRNA vaccines. To date, ARC-154 is the only siRNA vaccine candidate to receive regulatory approval. Despite this, siRNA vaccines have demonstrated significant breakthroughs both across infectious and non-communicable diseases. They elicit robust antigen-specific immune responses and can achieve protective immunity at lower doses compared to conventional vaccines, highlighting their potential for broader and cost-effective application. Therapeutic vaccines is another advancement where it treats existing diseases including cancer and autoimmune disorders by modulating the immune system rather than preventing the infection. In cancer immunotherapy, they aim to stimulate the immune system to recognize and eliminate malignant cells by targeting the tumor-specific or tumor-associated antigens. An example is the Provenge, the first FDA-approved therapeutic vaccine for prostate cancer. However, its use is limited by high cost and low immunogenicity. Recent advancements, particularly the saRNA platform, offer significant improvements as these allow for simultaneous expression of multiple TAAs or patient-specific neoantigens, triggering potent humoral cellular immune response while reducing the risk of tumor immune escape. Additionally, they offer scalability and cost-effective, making them more accessible than traditional personalized vaccines. Figure 9 represents some of the various cancer types that have been investigated in siRNA vaccine studies, where they have been shown to stimulate robust antigen-specific T cell responses against malignant cells. In the context of autoimmunity, where the immune system erroneously attacks self-antigens, tolerogenic vaccines are being explored. These vaccines aim to restore the immune tolerance by delivering disease-specific autoantigens or peptides, thereby suppressing autoreactive T and B cell responses, reducing inflammation, and minimizing tissue damage. Multi-omics integration has become a cornerstone of vaccinomics, aiming to understand the variability of vaccine response and accelerate both vaccine development and validation. By combining various omics studies, researchers can generate a comprehensive immune profile and identify biomarkers linked to immunogenicity, durability, and safety. For instance, genomic studies help to identify genetic variants affecting vaccine responses, while transcriptomic and proteomic data reveal real-time immune activation and cytokine patterns. Machine learning is often applied to these datasets, enabling the creation of predictive models that guide rational antigen selection, adjuvant design, and personalized vaccine strategies. Additionally, adversonomics, a subfield of vaccinomics, uses omics tools to investigate rare or severe vaccine-related adverse events, improving overall safety profile. Altogether, vaccinomics represent a data-driven evolution in vaccinology, making future vaccines not only more effective, but also safer and tailored to individual and population-level needs. Vaccinomics thereby lay the foundation for precision vaccinology. Traditional vaccines show variable efficacy across different populations, largely due to the differences in immune response and antigen clearance. Precision vaccinology aims to address this by achieving global health equity by ensuring fair access to vaccines and truly effective for all. This approach optimizes immunization strategies by tailoring adjuvants, antigens, and delivery systems to unique immune characteristics. Leveraging insights from multi-omics data, precision vaccinology uncovers distinct immune signatures that guide the design of personalized vaccines. Hence, the goal is to maximize immunogenicity, enhance safety, boost equitable protection worldwide, and accelerate responses to emerging outbreaks. Figure 11, as aforementioned, illustrates the precision vaccinology approach, which designs adjuvanted vaccines tailored to the unique immune profiles of vulnerable populations. By addressing the phenotypic and mechanistic immune differences, this strategy aims to optimize vaccine efficacy across all age groups. Key elements include selecting modifying adjuvants for enhanced effectiveness, optimizing antigen adjuvant formulations, and choosing the most suitable administrative route. This targeted design ensures robust and protective immune responses, a crucial factor for epidemic and pandemic preparedness. Lastly, let's dive into the vaccine clinical trials. Vaccines must undergo a rigorous developmental process, starting with the preclinical testing, where the target antigen is identified and the vaccine's efficacy and safety are evaluated in a lab and animal models. Phase 1 trials then involve a small group of participants and focus on the testing its safety and optimizing dose, typically taking at least a year. Phase 2 trials then expand hundreds of participants to assess immunogenicity and safety, ensuring both humoral and cellular responses are effectively stimulated. Phase 3 trials involve thousands of participants and test the vaccine's efficacy and safety in real-world conditions, often taking at least several years. Data from all phases are then reviewed by the regulatory agencies to determine if the vaccines meet the standards for approval. Finally, vaccines enter the post-marketing surveillance, involving continuous monitoring in the general population to detect rare or long-term adverse effects, assess real-world effectiveness, and ensure ongoing safety across diverse populations. Let's now take a look at some recent examples. Influenza is a highly contagious respiratory illness that causes significant morbidity and mortality worldwide. Due to its high mutation rate and antigenic drift, influenza strains constantly evolve, making it essential to update vaccine composition annually. This creates a persistent challenge in achieving optimal vaccine efficacy. To address this, Moderna developed the mRNA 1010, a next-generation quadrivalent mRNA influenza vaccine. This vaccine targets 4 key influenza strains, encoding hemagglutinin surface glycoprotein, aiming to elicit robust immune responses. It is currently in phase 3 trials, consisting of 3 parts, each evaluating the vaccine's efficacy and safety in different age groups, where each participant received a 50 microgram dose. The results demonstrated superior hemagglutination inhibition titers compared to the high-dose traditional influenza vaccine, indicating a potential stronger immune response. Although participants receiving mRNA 1010 reported more frequent solicited adverse effects, such as injection site pain, no safety concerns were identified. Human herpesvirus, also known as cytomegalovirus, poses a serious risk to immunocompromised individuals and newborns infected in utero. Despite its clinical significance, there is currently no approved vaccines, highlighting a critical gap in preventive care, particularly for seronegative women for childbearing age. To address this, Moderna developed the mRNA 1647, an mRNA-based CMV vaccine that encodes 2 key immunogenic components, glycoprotein B and the pentameric glycoprotein complex, both essential for viral entry and immune recognition. This vaccine entered the phase 1-2 trials in 2017 and enrolled approximately 154 healthy individuals, each receiving a single 100 microgram dose. Results showed a significant increase in the neutralizing antibody levels, along with the activation of CMV-specific CD4+ and CD8+ T-cell responses. Importantly, these vaccines were well tolerated, with all adverse reactions reported as grade 2 or lower, indicating mild intensity. Now, let us shift focus to cancer vaccines. One such example is a personalized mRNA vaccine developed to target melanoma. This vaccine encodes up to 34 patient-specific neoantigens, enabling the immune system to mount a tailored attack against each individual's unique tumor profile. It's administered intramuscularly using LNPs for efficient delivery. It has entered phase 2b clinical trials and involved 157 patients with completely resected stage 3b to 4 melanoma, all of whom received the vaccine in combination with pembrolizumab, a PD-1 immune checkpoint inhibitor. According to the latest update in 2024, the combination showed a notable benefit, where 2.5-year recurrence-free survival rate was 74.8% for the combination group, compared to the 55% for those receiving the monotherapy alone. Additionally, the overall survival trend favored the combination group, with 96% survival versus the 90.2% in the monotherapy group. With that, we can conclude that vaccination remains the most effective and cost-efficient strategy in combating infectious diseases. It continues to serve as a critical line of defense against the pandemic and emerging global health threats. While challenges such as suboptimal immunogenicity persist, innovations like siRNA vaccines are opening new possibilities, enhancing safety, immune responses, and scalability. These innovations also extend vaccine applications beyond infectious diseases, including oncology and personalized medicine. Moving on, continuous research and innovation driven by AI, systems biology, and interdisciplinary collaboration are essential to refine vaccine strategies and staying at the forefront of biomedical sciences. Ultimately, success also depends on strong public trust, awareness, and cooperation, making community engagement a vital part of the vaccination journey. These are the references. Thank you for your attention, and I'm happy to answer any questions.